Thank you for joining me. I want to start today by saying that we are doing fantastic progress on this course. And today we are doing a kind of an interesting thing. We are almost taking a step backwards and returning to talk about DNA. You might remember from the earlier on this course when we were introduced to it briefly. But today we are really going to zoom in and focus on it in a much greater detail. So, just to remind ourselves, in our last chapter we talked about Gregor Mendel and how he believed that there were these minute particles that must be passed from the parents to the offspring. And nowadays we realize, of course, that these must have been genes that he talked about. And imagine this was long before we knew anything about them, or even about DNA. So that is pretty remarkable, I would say. And then, much later, we realized that these genes must be encoded in the DNA, which is then packed as chromosomes. And I believe that I added this beautiful picture on your last week's material. This is a scanning electron microscopy image of 2 chromosomes. Isn't that just beautiful? I mean, this is why I love biology. We are privileged to see some amazing things. But for a long time, we were unclear if it is actually the DNA or protein that is carrying the genetic message. And of course, these days, it is easy for us to shout out that it is the DNA indeed. But this is only because we happen to know it as a fact, as we have been taught so. But if you were Alfred Hersey, yes, like the candy bar, and Martha Chase, who are shown in this picture, how could you test it? And now that you have learned about how the scientific process works, remember we talked about that right at the beginning of our course, you should be able to come up with a study to test it. So, can you come up with a plan? It is actually surprisingly hard to do so, isn't it? So if you have learned nothing during this course, I hope that you have now this newfound appreciation to how hard it is really to do science. So, let's have a look at what Alfred and Martha did, because it actually was pretty clever. They made 2 patches of material that they used for their experiment. And they used the radioactivity to label things in each. Remember, we talked about this at the early portion of our course, how radioactive tracers are used to follow where things are in biological organisms. So, for the first batch, which you will see above as this pink solution, what they labeled was actually protein part of a virus. And viruses can look like many different things. This is one that people tend to know the best, known as bacteriophage. Basically, at this stage, you can consider that what they labeled was everything else but the DNA, which is held inside the head part of the virus, and it is marked with blue in this diagram. So what they did is that they took a cell, which you can see as this orange-brown structure that the virus attaches to in these 2 images on the left. And they let this virus infect the cell. And remember, in the first batch that you can see in pink, they had labeled the protein part of the virus. And on the second batch, which is shown as blue, they had labeled the DNA of this virus. And we know that when a virus infects a bacterial cell, it moves its genetic material into the bacterial cell, and then this genetic material takes over the cell and makes it do all sorts of things that are good for the virus but bad for the host cells a lot of the time. So now we have 2 cells, both of them having been infected by foreign DNA. And now I'm certain that you can see where I'm going with this. Well, we put both mixtures into a blender and give it a good mix, and this mixing separates the bacteria and viruses. And now we run it all through a centrifuge, which separates the cells and viruses. And then we can study the cells only by extracting only those from this centrifuged mixture. And what we see is quite clever. We see 0 of a radioactively labeled protein inside the cell on the upper pink patch. And this is although we know that there is new DNA in our cell, because that cell is going all funny. But when we look at the lower series of pictures, we see that the patch 2 shows indeed that when DNA was labeled, we can now see the same DNA which was originally outside the cell, now inside the cell. So it must be the DNA that carries the genetic information. How clever is that? And actually what you see here is a real experiment where just that was done. And we can see that the DNA stained in yellow inside the head of a virus. And we can also see some of it inside the cell where the virus has squirted it inside too. So that is how we first discovered that DNA really carries the genetic information. Now, let's move onwards and have a little closer look of DNA. And some of this will be review of the stuff that we have earlier talked about. So, deoxyribonucleic acid, known as DNA, was this double helix structure. So you might recall me describing it as a long step ladder that has been twisted around itself. And it was made of nucleotides. And if you think of this step ladder, it is the sides of it that are made of sugars and phosphates. And then the horizontal bars that you step on when climbing the ladder, those are our bases. And there were 4 different bases. And let's have a look of them. So, the bases are marked inside this red box on the left-hand side, and ignore the uracil for now. So we have adenine, which binds with thymine, and guanine, which binds with cytosine. And their binding is very important. So just to remind ourselves, adenine and thymine go together. And guanine and cytosine go together. And how we actually found this out is kind of cool. When studying many different species and how many different individuals, we keep finding out that the amount of A and T is the same. And G and C is the same. Kind of cool, huh? And then this was further confirmed by many other experiments. And this is something that we call Chargaff's rule. Basically, it says that the amounts of adenine, thymine, guanine, and cytosine are different from one species to another species. But it always is the case that the amount of adenine is the exact same as the amount of thymine. And the amount of guanine is the exact same as the amount of cytosine. Isn't that just awesome what we can do with science? So the next thing that I want to talk about is something that I'm quite passionate about. And unfortunately, I believe that we do not teach enough about this to our students in a normal biology class, but I always try to sneak it in whenever there is a good chance. And you remember Charles Darwin and Alfred Wallace. That was an example of a question in bioethics. Was what happened there correct or appropriate? And another good example is an example that we will talk about next. And actually you will notice that on our second last week, you will have some work to be done relating to bioethics discussion rather than having a traditional lab. It is my hope that one day we will be able to offer students a course in bioethics. In fact, I have spent many years in the university studying this, and much of my research has been involved with things that could have been issues involving with bioethics unless those issues had been carefully considered first before a decision to run a study was done. So, let's talk about the story of discovery of DNA. And this story goes back to early 1950s, when at King's College in London, a young lady called Rosalind Franklin studied the structure of DNA. And this is probably one of the most famous images that she produced. And you might be like, what's so special about it? But it really is everything. It is amazing. And I often get saddened that these days the kids know who Kim Kardashian is, but they do not know who Rosalind Franklin or Marie Curie were. But I guess that it just makes me a bitter old person. Anyways, to produce these pictures, Rosalind used a technique called crystallography. And what you see here is actually a DNA double helix as viewed looking from the top down. Remember this structure, and if you use a little bit of imagination, you can see the sugar phosphate sides forming the outer sides of this circle and nitrogen bases going across the structure. It really is quite beautiful and extremely remarkable that we can see something like this. But that really was not a main interest of Rosalind's research, so she did not think much about it. But two guys called James Watson and Francis Crick saw this and took this idea and ran with it. And it is them who usually get credited for having been first to discover the structure of DNA molecule. And here you can see them showing it with this metallic model. And hear them as older guys. So I guess that my question is, should Rosalind Franklin get some of the credit? Simply because she did not make a big fuss about what she had shown with her crystallography work does not mean that she was not the first one to really show it. Or at least should she get partial credit? Well, these are questions that I do not think that there are definite answers to. But I do think that it is important that we talk about these questions just like we needed to talk about Darwin and Wallace. And actually, Watson and Kirk won a Nobel Prize for their work. Either way, enough of my bitter nature. So we know now that DNA is this double helix structure. And we are going to talk about next, how is DNA replicated? Remember, when a cell is getting ready to divide, all the material in it is doubled, including the DNA. And this process is rather unique. What happens is that the two strands that make up the DNA molecule open up and separate. And a new strand is built to match each of the free ends of the bases exposed. And this is why it is so important that only certain bases bind to certain bases. It works as a blueprint for making this complementary strand. And what we find is that now each new DNA molecule made in this way contains one old strand and one new strand. And this is why we call this process semi-conservative. We conserve some of the original one. So it is kind of semi-old strand. And this process involves 3 stages. The opening of the 2 original DNA strands, known as unwinding. And the enzyme called helicase helps with that. And then we build the complementary strands matching the bases. And finally, these join together to form 2 new DNA molecules from one original one. And DNA polymerase and DNA ligase help with that. One more thing that I do want to mention before we finish is that what really happens in eukaryotes is that this process of replication begins at the number of sites at the same time. So it is very efficient and proceeds fast. And this is where we will stop for now. We do have a tiny bit more to go, so the next section is going to be about RNA. But now it is time for you to take a break. Let's continue soon again.